Today we are going to discuss the structure of gynoecium, fertilization process, and structure of female gametophyte. See that the structure of gynoecium. Gynoecium is the female reproductive part of the flower, right? And this gynoecium contains the three parts. One is the stigma, style, and the ovary. Stigma is the toppest part of the gynoecium, and a tube-like structure is called style, and the swollen bottom part is called ovary. Inside the ovary, when you observe that ovules are located inside the ovary. The number of ovules are vary in the different plants. Maybe in some plants flowers contain one ovule, some flowers 2 or 5 or 100 also they can present, so that the number of ovules are different from different plants, right? When you observe the ovule, inside the ovule you can find a embryo sac. Inside the ovule you can find embryo sac. This embryo sac is composed of gametophyte cells. Embryo sac is composed of gametophyte cells, right? This is what the structure of a gynoecium, the female reproductive part of the flower, right? Coming to the process of fertilization, and we discussed already the pollination. Pollination means transfer of pollen grains from anther to stigma. After the pollination, you see that this is the toppest part of the female reproductive part, stigma, right? See this one, on the top of the stigma you can find one pollen grain. This is what a pollen grain. Pollen grain, right? So pollen grains are very light-weighted particles of the male gametes. And how this can be stick to the surface of the stigma? The stigma surface can secrete a sticky substance which is containing the sugary materials which provide nutrients for the development of pollen grain also. Because of the sticky substances, the pollen grains can be attached on the surface, and it containing some sugary substances which provide nutrients for the development of or growth of the pollen grain. When the pollen grain has developed, it produces a pollen tube. This is what indicating the pollen grain is developing. This is what the pollen tube. The pink color structure you are able to see, this one is the pollen tube. Okay, while the pollen tube is growing, you can find inside the pollen grain there is a nucleus. That nucleus is divided and produce the two nuclei, right? And two nuclei you can find in the bottom of the pollen tube. Two nuclei are present here, right? And when you see the ovary structure, inside the ovary, you find that already there is a ovule. Inside the ovule we can find a embryo sac, right? So this is embryo sac is protected by some layers. Embryo sac is protected by layers. These layers which protecting the embryo sac are called integuments. Integuments are the layers protecting the embryo sac, right? When you see that the embryo sac structure, there are 3 plus 3, 6 plus 1, 7 cells and 8 nuclei are there. Actually, this was formed by the 4 meiotic divisions. Okay, after 4 mitotic divisions, 7 cells are formed. 1, 2, 3, 4, 5, 6 and 7 central cell. This is right. So this is the structure which is present inside the embryo sac, which is called female gametophyte. So we will see that the female gametophyte structure it is. When you zoom this one, the female gametophyte structure, the female gametophyte containing 7 cells. The 3 cells are moving to the top of the female gametophyte, and 3 cells are coming to the lower side. Lower side is called this side is called micropylar end. Micropylar end. After 3 mitotic divisions, the 7 cells are formed here. The 7 cells, 3 cells are going to the chalagal end. The other side of the ovule is called chalagal end. This is called chalagal end. chalazal end. Right? 3 cells are moved to the chalazal end. The cells which are moved to the chalazal end are called antipodals. The 3 cells moved to the chalazal end are called antipodals. And there is a one cell at the center. It can move to the center of the female gametophyte, which is containing the polar nuclei. 2 nuclei are present. This is called central cell. Central cell, because the position which indicating that this cell is present at the center of female gametophyte. So it is present in the center containing 2 nuclei. So it is those are called polar nuclei. So remaining 3 cells they are coming to the or move to the micropylar end. The 3 cells present at the micropylar end are the one central cell is called egg cell, and 2 cells either sides of the egg cell are called synergids. Right? The central cell is called egg cell. The 2 cells either sides of the egg cell are called synergids. Right? And after fertilization, when the pollen tube is enter the embryo sac, the 2 nuclei present in the pollen tube, 1 nuclei fuses with egg cell. Here it is a 1 nuclei out of 2 nuclei present in the pollen tube. 1 nuclei fuses with egg cell. So this is called a primary fertilization. Primary fertilization. Primary fertilization, right? So when the primary fertilization occurs with the 1 nuclei of the polar nuclei and 1 nucleus of the egg cell, resulting the primary fertilization. So primary fertilization resulting the formation of a fruit. Formation of fruit. Later one more nucleus is there. After this fertilization, the another nucleus enter the female gametophyte and fuses with polar nuclei. The 1 nucleus fuses with 2 polar nuclei, and that resulting the secondary fertilization. Secondary fertilization. Secondary fertilization. Secondary fertilization resulting the formation of a special kind of tissue which is called as the endosperm. Endosperm. Right? You see that one nucleus with the egg cell and another nucleus with the polar nuclei. When you observe that the fertilization process 2 times. So primary fertilization and secondary fertilization. So both of them we can togetherly called double fertilization. Double fertilization. So primary fertilization resulting the formation of a fruit, and secondary fertilization resulting a formation of a special kind of tissue which is called endosperm. Right? And after these fertilizations, 2 fertilizations, the ovary converted into fruit, whereas ovules are converted into seeds. Right? And seeds are furtherly